Good morning, everyone. My name is Johnny Cardenas, and I'm the Global Marketing Manager for the Pharmaceutical Business Unit at AB Science. I would like to welcome you to the second of a four-part webinar series that AB Science will be broadcasting in conjunction with Drug Discovery Today, and the title of this webinar is, How Has HRMS Technology Fundamentally Changed the Way We Study Drug Biotransformation and Disposition? It is my great pleasure to introduce and welcome today's two speakers, Dr. Gary Impey and Dr. Mingshe Zhue. Gary Impey received his Ph.D. in Analytical Chemistry from York University in Toronto, in Canada, in 1998, and quickly joined Apotex Pharmaceuticals, where he worked in the Analytical R&D department. His graduate work involved instrument design and development, which led him to join the team at AB Science, demonstrating key features and benefits of the various mass spec technologies to prospective customers. Currently, Dr. Impey is Director of the Pharmaceutical and CRO-MS Business Unit at AB Science. Part of Gary's role involves the application of mass spec technologies in all areas of the drug discovery and development cycle, helping to guide the product development cycle with strategies to bring better solutions to the pharmaceutical marketplace. Dr. Mingshe Zhue is a drug metabolism researcher at Bristol Myers Squibb in Princeton, New Jersey, United States, where his teams have supported lead optimization of several discovery programs, preclinical studies of over 15 drug candidates in development, regulatory approval of Abilify, and DNA submission of dapagliflozin by providing ADME data. His research interests include application of LC-MS technology, optimization of ADME properties, metabolic activation, and regulatory drug metabolism. He completed his Ph.D. training in analytical toxicology at SUNY, Albany, and postdoctoral fellowship in drug metabolism at University of Washington. He has been frequently invited to speak at conferences and workshops, teach short courses at EAS, ACS, CPSA, and ASMS meetings. Dr. Zhue co-authored over 60 public research publications and co-edited 2 books entitled Drug Metabolism in Drug Design and Development and Mass Spectrometry in Drug Metabolism and Disposition. Before we begin, I would like to go through a few logistics with our webinar attendees. You should be seeing the main screen with 3 tabs displayed along the top of the main slide screen. These tabs are labeled as follows. The Questions tab. Following the presentation, there will be a question and answer session. You can input your questions via this tab throughout the event, so please feel free to ask any questions that you feel are relevant. The Feedback tab. We would appreciate you taking a few minutes to provide some feedback on our webinar. And the Support tab. If you're experiencing any technical difficulties during the webinar, please use this tab to gain assistance. Again, I would encourage you to input questions when you think of them, and these will be addressed in the question and answer period at the end of the presentations. The more questions that are asked, the more you stand to benefit from this event. Without further hesitation, I would like to hand over to Gary Impey to begin the webinar.
Great. Thanks, Johnny. Thanks for the introduction, and welcome, everyone. I'm glad you can join us today, and I hope that you'll join us for follow-up sessions. As Johnny mentioned, this is actually the second in a 4-part series of live webinars focusing on drug metabolism. And at each installment, we'll have a key opinion leader to share their experience and thoughts on drug metabolism workflows and the LC-MS technologies that they use to accomplish their goals. And we're proud to today have Dr. Mingshe Zhue with us. So just to kick things off, we thought it would be somewhat helpful to introduce this 4-part series and highlight some of the key topics that will be covered. We also thought it appropriate to follow the various stages of the drug discovery and development process with each installment, covering a different portion of the pipeline and the key drug metabolism applications that take place. So with that, I'd like to step you through some examples of topics that will be covered throughout this 4-part series. And please keep in mind that we're now on the second installment of this 4-part series. So in the early discovery phase, the simultaneous metabolic stability and soft spot analysis is really a good example of the challenges of performing both quant and qual workflows for pharma. We need high throughput relative quant information on the parent drug of interest, while at the same time getting identification of the major metabolites. And this typically happens under high throughput LC conditions. The TOF 5600 simplifies this data collection and analysis. A simple generic TOF-MS scan is followed by automatically acquired MSMS on the major metabolites, and the accurate mass helps to provide structural information. And accompanying software tools like Multiquant and Metabolite Pilot ultimately help with the overall approach. Staying in the early discovery phase, another important aspect of drug metabolism is the presence of GSH conjugated metabolites. This is an indication of the formation of reactive metabolites, and it's pretty critical to identify and monitor. The QTRAP 5500 is really the first LC-MS system that combined the most powerful modes of GSH metabolite detection into a single injection workflow with sufficient speed and sensitivity. You can monitor multiple precursor ions and or neutral loss scans along with polarity switching and collect MSMS in a single injection. So as we move to later stages of discovery, we begin to work with much more complex samples. We still need to identify and characterize metabolites, but the added complexity requires us to either collect information differently or use advanced LC-MS technologies and innovative workflows that simplify that analysis and ultimately help us retrieve the information that we're looking for. So along with the performance capabilities of the TOF 5600 from a hardware perspective, there's also some innovative tools for MetID, including multiple mass defects filtering and a unique neutral loss mode. These techniques provide a much more efficient way of getting information on relevant peaks of interest versus just collecting a lot of MSMS data in hopes of retrieving it from complex software analysis. So while these workflows, implement some kind of targeted or semi-targeted acquisition, you can also simultaneously collect non-targeted information that can be, queried for those unexpected metabolites. When sensitivity is the most critical, especially when dealing with very complex, dirty samples, the QTRAP 5500 platform offers the predictive MRM approach. Here's a targeted list of MRM transitions that are used to screen for metabolites, and MSMS information is collected in the same analysis. It is a targeted approach, but the flexibility of the system also allows variations of or to this workflow, if you will. The MIMS approach, or MIM, is a great example of this. Instead of selecting fragment ions for the MRM screen, a simple parent-to-parent transition will capture all species across a very large mass range from one AMU to the next. So no knowledge of metabolite fragmentation is actually required. A great system to apply both targeted and non-targeted workflows together. Now, in development, we need definitive ID on all the metabolites for the lead compounds. Accurate mass is required. The TOF 5600 provides that capability. So to bring this brief intro to a close, the TOF 5600 and the QTRAP 5500 are really two complementary tools that help you integrate qual-quant workflows in your lab. Whether it be time course studies or cross-species comparison and quantification of metabolites, these two platforms improve efficiency, help you collect more information faster, and ultimately reduce irrelevant data collection and analysis. So with that, I will pass it along to Dr. Mingshe Zhue. Mingshe Zhue is a drug metabolism researcher at BMS in Princeton, New Jersey. And today he'll discuss how high-resolution mass spec technology has fundamentally changed the way researchers study drug metabolism. And thanks for your attention. Mingshe?
Yes. First, I'd like to thank the Drug Discovery Today and AB Sachs to organize the webinar. I also like to thank Johnny and Gary for the introductions. So it is my great pleasure to have such opportunity to share our experience in the use of the high-res mass spec in drug metabolism profiling. So what I'm going to talk about today, I'm going to talk about how the high-res mass spec technology has fundamentally changed the way we do drug material study in the industry. So in today's presentation, I'm going to talk about 3 parts. First, I will going to give you a brief introduction regarding of the challenge we face in drug material identification using the mass spec. I'll also give you some a brief description of the technology evolution in last 20 years, how we use the mass spec to do the drug material identifications. In my second part, we cover some details of the workflow use of high-res mass spec, as well as some algorithm and data process technology that are routinely used for the drug material identification use the high-res mass spec. My third part of the talk is a few examples that apply for recently introduced ABR 5600 high-res mass spec to do drug material identifications, which including 2 examples. So in this slide, in the middle of this slide, is a 4-scan spectra of data. Okay. So we very often see this kind of data acquired from animal or human species. You can see many ions presented as 4-scan spectra data. The question is, what is the metabolite ions? So basically, to identify molecular ion of metabolites in very complex spectral data set is the most challenging of task in metabolite identifications, especially when those metabolite ions have no way to detect by UV or radio detection technology, or their molecular weight are unpredictable. Because of these reasons, metabolite identification was a bottleneck of DMPK study in drug discovery back to 4 or 5 years ago. So as you can see from this slide, in drug discovery DMPK study, including metabolic stability study, P450 inhibition, permeability study, bioanalytical in PK study, as well as metabolite identifications. So usually, all those studies, except for metabolite, are involved quantification analysis use triple quadruple, which can be finished with 1 or 2 minutes. The productivity of those studies can be measured by compounds per day per person. So usually, those quantification analysis for metabolic stability study, one scientist can do 20 to 50 compounds per day. For P450 inhibition study, it's about 10 compounds or 30 compounds a day. For bioanalytical analysis, for PK analysis, a scientist maybe can finish by 5 to 10 compounds or even more compounds per day. However, if you look at the metabolite identifications back to the 4 or 5 years ago, which is really tedious work. Usually, per run we take 30 minutes. Maybe you need to use multiple injection or use multiple instruments. In general, a scientist can finish the metabolite identification 1 or 2 compounds per week. So by that time, metabolite identification is truly bottleneck of the DMPK study. Things have been changed in the last few years. So right now at Bristol Myers Squibb, we can do, a scientist can finish the metabolite identification for 5 compounds per day in average. So what happened in the last several years? In order to understand what we are now, we have to understand what happened in the future or in the past and what could happen in the futures. So the HRMS technology truly used in industry is about 20 years ago. That time, electrospray HRMS are available. 10 years ago, in the industry, people use 3 different technologies to do metabolite identifications, which including the triple quadruple, untrapped technology, and the high-res mass spec. So we call the combination of those technologies is a classic approach for metabolic identification using HRMS. in the approach, people use triple quadruple neutral loss scan or percussive ion scan to detect the unknown compounds. Then use untrapped technology to get multiple MS2 spectral data. In some cases, people use the high-res mass spec to determine the molecular formula of unknown metabolites. About 5 or 7 years ago, a lot of things have been changed, and a lot of things happen, including the new technology, new instrumentations, including Q-trap technology, orbitrap technology, and improved TOF technology. In addition to those instrumentations improvement, several data mining technology was introduced, including mass defect technology, as well as the MSE or other data acquisition technology was introduced. Because of those changes or innovations, so right now people actually use a single LC-MS platform to do drug material identification and quantifications. So mainly including the Q-trap and the high-res mass spec. So in today's presentation, I only focus on the high-resolution mass spec. So now I use one slide to overview what is the high-res mass spec technology used in drug material identifications. So let me walk through slowly. As you can see from this slide, high material identification uses high-resolution mass spec divided by two steps. First step actually is the data acquisition. Use the data-dependent acquisition method, which including data-dependent acquisition, use the list-dependent data acquisition, isotope-dependent acquisition, or the MSE technology. So those kind of data acquisition allows us to have two sets of data. One is called full-scan mass spectral data. Another is the MS2 spectral data. So this is the first step of the analysis. The second step, usually we can do at the same time, is called the data mining. So that there are several data mining tools allowed us to use, including extract the unchromatographic analysis, which targeted metabolites with no molecular weights. Second technology is called mass defect field, which is targeted metabolites based on the predicted mass defect values. Another technology we use in some cases is called isotope pattern filtering, which is a search for metabolites which have unique and distinguished isotope patterns. Another algorithm we used in many cases is called background subtraction or control sample comparison, which allowed us to detect metabolites that are not presented in control samples. So all the four of those data mining tools allowed us for process the full scan spectral data set. Additionally, there are additional tools, including the product time filter or the neutral loss field, which can be used for process the MS2 spectral data to find the metabolites. So thus, once you identify the molecular ion for unknown metabolites, then you can recover MS2 spectral data from the MS2 spectrum. So based on the molecular ion and the MS2 spectral data, we can come with the metabolite structure. Okay. So in next part, I'm going to talk about a general workflow as well as the common data process algorithm that are for the metabolite identifications. First, I would like to introduce the mass defect filter. The mass defect filter is the first technology was developed for the high-resolution mass spec for metabolite detections. Actually, which was developed by the scientists at Bristol Myers Squibb back to 7 years ago. A scientist at BMS is called Dr. Zhang Haiying. He found out a very interesting phenomenon. So what he see is that he found that the metabolites shift of the molecular weight of metabolite shift from parent drug are significant. However, the mass defect value of the metabolite shift from parent drugs are in very small range, which is plus or minus of 50 millidalton. For example, hydroxylation metabolite molecular weight of shift is about 16 mass units, but their mass defect change is very small, is about 5 millidalton. Demethylation metabolite, their mass defect change is only about minus 23 millidalton. Therefore, he thinks we can use this phenomenon to specifically extract metabolites from the full-scale spectral data acquired by higher-risk mass spec. So basically, the mass defect filter is a post-acquisition process that retains metabolite ions have similar mass defect values to those of the parent drug or their structures, core structures. As a result, interference ions that have significant different mass defect are filtered out. Now I use a few examples to demonstrate how the mass defect works. So this slide shows the mass defect workflow we are commonly used in drug metabolite identifications. So the first step is acquire full-scan spectral data using any high-resolution mass spec. Then you use multiple mass defect filter to filter out the interference ion to detect the different class of metabolites. If you want to find the oxidative metabolites, their structures are similar to the parent drug, you can use is called the drug metabolite filter. If you want to detect metabolites which are very smaller than parent drug, you can use core spectral or core structure mass defect filter. If you want to detect the conjugated metabolites, for example, gluconjugate, you can use gluconjugate mass defect filter. So, for example, this top panel is the mass defect process profile from the full-scan spectral data collected from analysis methadone incubation in human liver microsome. So what we can see is several metabolites are in addition to the parent drug. All those metabolites have similar structure to the parent drug. Their mass defect value very close to the parent drug. The bottom panel actually is the TRC of the mass defect process profile use a combination of the multiple mass defect filter. All oxidative metabolites, including M13, which is metabolite significantly smaller than parent drug, actually is in the alkylated metabolite also detected by this process. So this is a table summarizes the metabolites of nifedipine, which is detected by use of the nifedipine as the mass defect filter template. As you can see, all those metabolites have several molecular weight to the parent drug, including the monohydroxylation metabolites, dihydroxylation metabolites. Most interestingly, have one metabolite called M4. Its molecular weight is 12 mass units smaller than parent drug. Actually, it is the uncommon metabolite. The mass defect factor can clearly detect the uncommon metabolites, regardless their molecular weight or fragmentations. So now I can use detailed information to illustrate why mass defect technology in conjunction with the high-resolution mass spec is better than a neutral loss skin or percussion skin. So this is the structure of the nifedipine. Nifedipine under the CRD undergo two major fragmentations, including formation of the product ion of the 246 by loss of neutral species 224. Another fragmentation is the formation of the product ion 274 by loss of the 196 Dalton. So this is the nifedipine spectra. So nifedipine catalyzed party for 50 generated several major metabolites, including hydroxylation of this metabolite and the hydrolyzation metabolites of the M2. All those metabolites have unique fragmentation similar to the parent drug. Therefore, either neutral loss or precursor ion scan can detect those metabolites. Nifedrazone also generates M4. This is an uncommon metabolite. Their molecular weight, as I mentioned, is smaller than parent drug, is about 12 mass units smaller than parent drug. Okay. All those three metabolites are detected by triple quadruple use neutral loss scan or product ion scan. Okay. But neutral loss scan or precursor ion scan cannot detect the M13, which is the NDS-alkylated metabolite of nifedrazone. You know why? Because the nifedrazone spectra of the fragmentation of the nifedrazone are significantly different from the fragmentation of this metabolite. Therefore, neutral loss or precursor ion scan did not work with this metabolite. In all these cases, mass defect filter are able to detect all those metabolites because the mass defect is not dependent on fragmentations. It depends on predicted mass defect values. So now I'm going to talk about another data mining tool which is commonly used for high-resolution mass spec analysis. We call this is called the isotope pattern filtering. So we use this technology routinely for rectal metabolite screen. So this slide shows the workflow of use high-resolution mass spec in conjunction with the isotope pattern filtering for detection GSH adduct. What we do is here in the incubation, we're including the testing drug, a human liver maximum, as well as a mixture of the GSH and the stable isotope labeled GSH. At a ratio about 1.1, 1 to 1. The mass units difference between GSH and the stable lesser GSH is about 3 mass units. So after we generate the incubation sample, then inject it to the high-resolution mass spec. Then we use it's called isotope-dependent mass MS2 data acquisition to acquire that. As a result, the acquisition could generate 2 sets of data. One is the 4-scan spectral data, which allows the process to use the isotope pattern filtering to specifically target the GSH adduct. Also, the isotope-dependent acquisition are selectively to acquire MS2 spectral data for those species that show specific isotope pattern. So once we identify molecular ion of the GSH adduct, then we can recover the MS2 spectral data in the MS2 spectral data set, and based on the mass spectral data, we can come up with the spectrum, the structure of the GSH adduct. So now I'll show you one example. Use the high-resolution mass spec and the isotope filtering to detect the GSH adduct sensitively. The compound tested in here is a diclofenac acid. We incubated the diclofenac acid 10 micromole in the liver maximum. After 30 minutes, in the presence of a mixture of the GSH and a stable labeled GSH. So this is a profile of the 4-scan spectral analysis of the samples. As you can see, all those peaks are not related to GSH adduct. They are either endogenous components or oxidative metabolites, not a GSH adduct. After the process of those data sets use the isotope pattern filtering, we are able to remove all the interference ion to detect the GSH adduct. In this profile, what we can see are all GSH adducts as no positive components. So based on those data, we are able to come with a molecular weight of the GSH adduct and the elemental composition of the mass of these reactive metabolites. So all the data process just take a couple minutes. This is one example really elucidated the sensitivity, speed, and the selectivity of those technology in the reactive metabolite screen. The third algorithm we are used very often in conjunction with high-resolution mass spec, we call it background subtraction technology. So this technology, one of the algorithms used in our process, I was developed at BMS. The principle behind of this process is very simple. As you can see, we have two sets of data acquired from the high-resolution mass spec. One is the dose sample. In the sample, you can see there are a lot of endogenous components, interference, but we have seen several data and actual, those are the drug-related components. Another set of data is a sample collected from pre-dose samples, either from plasma or urine. So all those samples are including all the interference and endogenous components, but no drug-related components. If you subtract those set of data from the pro-dose sample, what does remain? Remain actually is the metabolite ion or drug-related components. The principle is very simple. Now I'm going to show the effectiveness of those technologies, which was presented by Han Ying a couple years ago at the HRMS. The sample we tested is called retinoidine. Actually, we dose those samples as a compound in the human and in the about the 150 meg dose in humans, and then just collect the sample in plasma after dosing or before the dosing. The top panel actually is the TRC of a full scan analysis of the sample after the 6 hours of the dosing. All the peaks shown in the top panel actually are endogenous components, except the parent drug ion here. But the bottom panel is the background subtraction process file. You can see all the endogenous components are removed, remain actually are drug-related metabolites. If you enlarge those parts, you can see in the bottom line, we are able to see some minor peak, which actually are metabolites. You can see here, all those minor peak actually is metabolites. In addition to the facility of the detection of metabolites in complex biological matrix, the background subtraction allows us to clarify the spectral data to find out what is the molecular ion. So the top panel is unprocessed full-scan spectral data. You can see most actually are the endogenous components. The bottom panel are processed full-scan spectra. All the endogenous ion have been removed, remain actually the drug-related components. So you can see here, so actually based on those two ions, we come with a structure of those new metabolites through the uncommon biotransformation pathway. So I just talk about the second part, how to use the basic workflow and the basic algorithm to do metabolite identification, which including the mass defect technology, isotope fitting technology, as well as the background subtraction technology. I think right now most of high-resolution mass spec instruments, including those algorithms, all those algorithms can be used together or separately. It really depends on what you want, what is available. Now I'm going to move to my last presentation, application of a triple quadruple or triple top 56 instrument in the analysis of the metabolites. So for example, I've been talking about application of this technology in the analysis of diachronic metabolites in BDC rights. As I mentioned earlier, there are several technology, instrumentation, and algorithm which allowed us to do fast, high-throughput modifications, especially for in vitro sample. As illustrated in this slide, sample can be injected to the high-resolution mass spec, and then you can use the variety of data acquisition, including MSE, isotope pattern dependent acquisition, or the intensive visualization to collect both full-scan spectral data and MS2 spectral data. Then you can use the different data mining tool, including mass defect filter, isotoping filter, to find the molecular ion of mass defect, molecular ion of unknown compounds. Then you can recover the mass spectral, MS2 spectral data from what you collect, the MS2 spectral data, and come with the structure of the metabolites. But however, all those technology only works on beautifully for in vitro sample, because the data-dependent acquisition, including the MSE technology, are not fully workable for in vivo samples, especially those samples have a lot of endogenous components. Isotope pattern-dependent acquisition only work for compounds which have very specific isotope patterns. Most of the drug in vivo metabolites do not have those isotope patterns. The intensity-dependent acquisition are not able to target the metabolites if their intensity are very low. So therefore, we need to find an additional way to acquire the MS2 spectral data to high throughput modification of in vivo metabolites. So once we see the capability of the 5600, we are very happy, because the 5600 have unique algorithm, unique data acquisition technology. We call this the mass defect-dependent information data acquisition, which allowed us to specifically target the metabolites based on their predicted MS2 spectral data. So this slide show the workflow we used for analysis in vivo metabolites. Use the 5600 instrument as well as the mass defect-dependent acquisition. So as you can see, so we have the in vivo sample, then inject the trouble top, then we use the mass defect-dependent acquisition, collect the full scan spectral data as well as the MS2 spectral data. Then we use the mass defect of extract ion chromatography analytical analysis to process full scan spectral data to find the metabolites, then back to the MS2 spectral data to recover the spectral data of no metabolites, then come with the structure of the metabolites. The compound we tested is a diclofenac acid. Basically, we inject the diclofenac in the BDC rice and collect the biurine and do the sample analysis, sample process, then we injected to the 5600. The top panel is the full scan spectral data of the biosample. So you can see most of the peak actually come from endogenous components. What you can see only major metabolites and parent drug. The bottom panel actually is mass defect process profile by using parent drug as the mass defect template. All those metabolites are oxidative metabolites. The structure are very similar to the parent drug. So, for example, in the luteation time, 17 minutes, there is a very minor metabolite is M12. So the mass defect-dependent acquisition allowed us to specifically target this metabolite to acquire MS2 spectral data. So this is the full scan spectral data M12. So you can see we don't see a major ion in the spectrum, actually the endogenous components. If you enlarge this part of the spectrum, you do see few metabolite ions, so that which is very close to the background. Those two metabolites, including the 294 and the 312. Actually, the isotope mass defect dependent acquisition really can track the MS2 spectral analysis for these two metabolites. So this is a spectra show the MS2 spectra of the MS2 metabolite M12, which acquired by mass defect dependent acquisition. So the spectra is very clean, was able to include find even very minor fragments, which is very important to allow us to identify which moiety actually the hydroxide group located. So that in same principle, we can process the full scan spectral data acquired from urine sample. So top panel actually TRC of the urine sample. So you can see most of the components actually from the background from the endogenous components. As you can see from top panel, the bottom panel is the mass defect process profile. All those are oxidative metabolites presented in urine sample. So this table, to summarize the oxidative metabolites, we analysis from the urine and the bile. So as you can see, multiple metabolites are found. All those metabolites actually were triggered by MS2 acquisition. So basically, the technology really allowed us to one injection get both of the MS2 spectral data and the full scan spectral data. As I mentioned earlier, the full scan spectral data not only can be processed using a variety of mass defect templates. If we use the group conjugate template, which will allow us to find the group conjugate in the data set. The top panel is a group conjugate mass defect process profile. All you can see is a group conjugate presented in the bile. The bottom panel is a group conjugate mass defect process profile from the urine sample. All those metabolites are group conjugate presented in the urine. So another example is the process of the use of the group conjugate or GHG adduct as the template. All we can see here is a multiple GHG adduct detected by mass defect dependent acquisition, and some of actually the GHG adduct are newly identified GHG adduct. For example, two metabolites which is GHG adduct very minor, including the M10 and the M18. So one of the things I like you pay attention is the very minor GHG adduct at molecular weight of the 617. If you look at the spectra, full scan spectra of data, the molecular ion of this group GHG adduct actually almost close to the baseline. So this is the molecular ion 617, but the mass defect dependent acquisition allows us to selectively target the spectral MS2 spectral data. So this is for MS2 spectral data acquired by mass defect dependent acquisition. Based on the spectral data, we are able to come with the structure of those very minor and GHG adduct. So in comparison, if we use the intensity dependent acquisition, we are not able to acquire the GHG adduct. Those technology only acquire the MS2 spectral data of most intensive ions. So another example, if we use the dynamic background subtraction technology, which is good for most of the components, but that technology was not able to acquire MS2 spectral data of this very minor GHG adduct. So this table summarizes the data acquired by three different data dependent acquisition. So as we can see in the bile sample, there are 31 metabolites, including the parent drug. The mass defect dependent acquisition able to acquire MS2 spectral data for all those 31 metabolites. Intensity-dependent acquisition was able to acquire about 50% of the metabolites. DBS, IDA are in the between. So those really illustrate the selectivity and sensitivity of the mass-dependent acquisition for analysis in vivo sample in very complex biological matrix. So the final example I think to illustrate is the application of the TOF 5600 in analysis of the multiple components in P450 inhibition cocktail assays. So as we know that high risk is the mass HRMS platform choice for metabolite ID. Okay? But how about can high risk mass spec be used for other drug metabolism tests? For example, metabolic stability study, PEMPA protein binding analysis, protein reaction phenotyping, plasma metabolic quantification. Yes, all those quantification work associated with drug metabolism study are being used with the high-risk mass spec, has been reported recently, but not the P450 inhibition study use the cocktail approach. Cocktail assays in P450 inhibition really represent the challenge of the quantifications. There are several reasons, because in cocktail assays, multiple components, they have different devoltified structure. Some metabolites actually have very low concentration. Some metabolites are not sensitive to the electron spray. So the objective of this study is to develop the methodology for P450 cocktail assays. So from those studies, we want to demonstrate that the high-risk mass spec can meet the challenge in quantification analysis. The cocktail assay we used is we selected 5 P450 probe substrate, including the 2C19, 2D6, 3A4, 1A2, and 2C19. All those are major P450 take a major responsibility for the drug clearness in most of the marketed drugs. So first we saw that we can use the high-risk mass spec data for scan spectral data. Then based on their molecular weight, we can see. But however, one of the analysts, which is a metabolite of methylphenidone, actually the concentration of this was very low. As you can see in the top of panel, in the incubation, we do able to see the metabolites. However, in the presence of the 2C19 inhibitor, we are not able to use the full scan spectral analysis to detect the metabolites. As we know that the TOF 5600 have very unique scan function is called the high resolution the multiple reaction monitor. In this case, we're able to select the molecular ion in Q2. You get the MS2 fragments in Q2, and we then monitor the product ion in Q3. Therefore, we use this specific data acquisition to quantitative analysis this unique metabolite. We found that the sensitivity was significantly improved. As you can see in the top panel is the metabolites of the methylphenidone. The middle panel actually is the metabolites which are presented in the presence of the 2C19 inhibitor. So metabolites even in the low concentration was clearly detected by this method. So therefore, we came with the approach, basically we use a combination of the full scan analysis and the multiple reaction monitor to detect the 5 metabolites of the probe substrate. So this is to show you the each individual metabolite, which are well separated by 5 minutes run, and the synthetic metabolite was monitored by high resolution MRM analysis. A rest of metabolites are monitored by full scan analysis. As a result, we are able to detect the acquire the IC50 for the classical P450 inhibitor. The data are very consistent with the data reported in the literature. So those examples really illustrate a high-resolution mass spec in conjunction with the multiple data acquisition methods really can meet the needs for quantifications. So now I'm going to end my presentation by using a summary. As we can see from the data presented, high-resolution mass spec together with the new data mining tool has become the HRMS platform of choice in drug metabolite identification. Triple top with mass defect dependent acquisition is a powerful tool for the in vivo metabolite identifications. Additionally, the high-resolution mass spec together with the different technology really can meet the challenge in the in vitro quantifications. So based on those data, we are very confidently to use the high-resolution mass spec in variety of DMPK study at Bristol Myers Squibb or other companies, which including one is the high throughput analysis of reactive metabolic screen, as well as the metabolic soft spot analysis. So we are able to analyze the compounds, probably 5 compounds per day. Another application is the drug metabolite analysis or dispersion in animals, as we demonstrated in this presentation. Another application is metabolite profiling and qualification in first-in-human study by use the background subtraction analysis. Obviously, we can use a high-resolution mass spec for lots of the in vitro ADME study, including the P450 inhibition, as discussed today, as well as the metabolic stability analysis, P450 reaction phenotyping. Finally, in many companies, we started to use the high-resolution mass spec for discovery bioanalytical analysis of small molecules as well as the proteins. When we use high-resolution mass spec, we can eliminate the needs of the method development, which is very useful in drug discovery. Finally, I'd like to thank our colleagues at Bristol Myers Squibb, as well as our collaborators from the AB sites, as well as in the Central University in China. Thank you for your attention.
Thank you very much, Mingshe. That was an excellent presentation. I can see that all of our attendees have been busy sending questions through for our speakers. So we have about 7 minutes left for a question and answer period. So Mingshe and Gary, if you're ready, why don't we begin with our first question?
Yes.
Mingshe, this question is for you. On the third slide in your presentation, where you described MetID as being the bottleneck with 10 to 30-minute run times and multiple injections being required, and only covering 1 to 2 compounds per week, how has this changed with the new workflows that you're using, like mass defect filtering?
I think this is a very good question. The slide actually I used was back to the 3 or 4 years ago. But right now, I think in few companies, including BMS and Merck, are able to use the high-resolution mass spec and a variety of data mining technology, including MSE, data acquisition, mass defect filter, or other technology. I think we are able to analyze the metabolites of compounds, probably about 4 or 5 compounds per day, per instrument, per scientist. So this is for data acquisition. Data interpretation really depends on expertise of individual scientists. With the availability of the data interpretation software, we can come up with the structure very rapidly. But in general, I think the productivity of the metabolization are tenfold increase, I think, compared with 5 years ago.
Great. Thank you, Mingshe. We have another question for you. Can you comment on which acquisition technique you relied on to find and ID metabolites?
It really depends on the purpose of the study, as well as the metrics we are dealing with. For example, if we're talking about high throughput metabolite ID, which is often used in drug discovery, including metabolic soft spot analysis, and as well as the reactive metabolic screen, we deal with a larger number of samples. In this case, we have to use the data acquisition, which is not dependent on compounds. It is so therefore, we have to use the data acquisition which is generically, okay? Such like the not mass defect dependent acquisition, it's such like the MSE technology. And isotope dependent acquisition, as well as the dynamic background subtractions, which are available in 5600. In those data acquisition, basically, you don't need to design any specific data acquisition method. It's not dependent on compounds. Okay, so this is one of our applications, high throughput analysis. The second important study we often deal with is the in vivo sample, which is very complex. The data dependent acquisition like MSE, isotope dependent acquisition, in most cases are not work nicely with the in vivo sample. So as mentioned in today's presentation, I think the mass dependent acquisition will be perfect choice to analysis in vivo samples, especially those samples have a low concentration of metabolites and in a very complex biological matrix.
That's great. Well, that was a great answer, Mingshe, and probably perfect for the next question that's coming up. Can you comment on the dynamic range of the system? Specifically, what is the spectral dynamic range of the system when dealing with low-level metabolites and major metabolites during high-throughput detection?
You mean if I understand the question correctly, you mean the dynamic ranges?
Yes.
Okay, dynamic ranges are very important in the quantifications. Okay, so in general speaking, high-resolution mass spec has a limited dynamic range. Usually is a threefold, okay, 10 to threefold. But with recently introduced technology, including I think the 5600, as well as like the benchtop, the Orbitrap, dynamic range of the data acquisition in full scan analysis achieve probably 10 to 4. Some cases came to the 10 to 5. Okay, so this is in general speaking. But I have to mention this: the 5600 have the unique scan function, including full scan analysis and the multiple reaction monitor. So those actually we are able to use the two scan function together, which I think tremendously increase the dynamic range for the quantification of analysis of the same components.
Great. Excellent. And I think we've got time for one last question, Mingshe. Given the results you're getting with the strategies you're using, how often do you go back and review data sets to reinterrogate them?
This is a very good question, sir. I don't think that any technology are perfect. Okay, I cannot say one technology is better than others. It really depends on how to use different instruments, different data mining tools to solve the special issues. Okay, for example, mass defect has limitations. Mass defect selectivity, sensitivity is dependent on the compounds, as well as depend on the matrix. Mass defect work nicely for plasma sample, biosample, but not the urine samples, because in a urine there are a lot of small molecules which may have same mass defect with the drugs. So this is one example.
Okay.
Therefore, in this case, very often we're going to miss the metabolites or get a lot of false positive. And the background subtraction is a great technology, but the background subtraction, in order to get perfect data, you have to use analytical control samples. Sometimes very difficult to get. Therefore, we could miss the metabolites. In all those cases, though, we need remassage the data, reprocess the data. The beauty of the high-resolution mass spec is that in many cases, we don't need to make additional injections. If you get full scan spectral data, or you get most of the MS2 spectral data, which really allows us to reprocess the data, get new information.
Fantastic. Thank you very much, Mingshe. I'm afraid that's all the questions we have time for. Thank you all for your participation and submitting some very high-quality questions. Any questions that we were unable to answer today due to time constraints will be passed on to our speakers, who will then try to contact you later on with an answer. I'd like to take this opportunity to thank Dr. Gary Impey and Dr. Mingshe Zhue for their excellent presentations, and to all our attendees of today's webinar. This has been a very informative session with a lot of thoughtful questions and answers and some very thought-provoking ideas. Please don't forget, there will be a recording of this webinar that will be available online very shortly. And also, please stay tuned for our third installment of this four-part series, which is tentatively scheduled for October 2011, and will feature a speaker from the greater European area. Thank you, everyone, and hope you all have a great day. Goodbye.